This is Lecture 5, where we will go over the prokaryotes. Prokaryotes also show a great amount of diversity in their nutritional adaptations. They have a broad range of feeding strategies. Some prokaryotes are considered autotrophs and can synthesize their own organic nutrients from inorganic molecules such as carbon dioxide and bicarbonate. They can also be classified as heterotrophs, which are prokaryotes that require at least one organic compound in order to manufacture additional nutrients. If you take a look to the diagram or chart to the right, we can see different types of organisms that are considered photoautotrophs, chemoautotrophs, photoheterotrophs, as well as chemoheterotrophs, meaning that the original source of energy is either light in the instance of phototrophs or organic compounds in the instance of chemotrophs. Some prokaryotes are classified as obligate aerobes, meaning that they require oxygen to carry out cellular respiration, while other prokaryotes are obligate anaerobes, meaning that they are actually poisoned by the presence of oxygen. These anaerobes would need to carry out fermentation or other anaerobic respiration pathways. Some of these pathways include using nitrates or sulfates as their final electron acceptors. Some prokaryotes are also facultative anaerobes, meaning that they can do both. They can use anaerobic pathways, but they can also utilize aerobic respiration when oxygen is present. Some prokaryotes can undergo nitrogen fixation. Nitrogen is considered to be an essential element for the synthesis of both nucleic acids and amino acids. Some cyanobacteria, as well as some archaea, like methanogens, can fix nitrogen gas into ammonia. This can then be easily incorporated into more complex organic molecules. These prokaryotes are very important in making nitrogen available for other life forms as they are consumed. It is important to understand that in many prokaryotes, a single cell cannot carry out many pathways at the same time. For instance, the oxygen produced from photosynthetic pathways will inactivate the enzymes that are actually required for nitrogen fixation, thus prohibiting those pathways from occurring at the same time. In order to overcome this, some prokaryotes are known to form filamentous chains. These chains have different cell types that can carry out different functions. For instance, in the chains depicted in the picture below, we can see some photosynthetic cells that would undergo the process of photosynthesis, whereas cells within the heterocyst would carry out nitrogen fixation. We can also see this cooperative behavior occur between different prokaryotes in the production of biofilms. In this instance, we have the cooperation of multiple prokaryotic species. Cells within the biofilm can secrete signals that can recruit nearby cells. Some cells can produce important polysaccharides and proteins to form capsules and also to increase the adhesion to both surfaces and other cells. And we can also see the creation of channels to help direct nutrients to the inner layers and also to allow for waste elimination, thus having the biofilms almost act as an organism. The formation of biofilms is very common in nature. However, they can at times be harmful. In some instances, biofilms can cause infections, can cause contamination of surfaces, instruments, and lenses, can be resistant to many different drugs, and it may also cause damages to surfaces as well as infrastructure. In Pseudomonas aeruginosa, this is a common bacteria that is found in nature and also in the human intestine. However, biofilms that contain this bacteria can cause very harmful infections that tend to be resistant to antibiotics. Metagenomics has had a huge impact on our understanding of prokaryotic diversity. Metagenomics involves the study of genetic material from environmental samples. It has been widely used to both collect and classify prokaryotes. Metagenomics has also led to major revisions in taxonomy. For instance, it has allowed us to conclude that archaea is more closely related to eukaryotes than bacteria. In addition, it has led to our understanding that gram-negative bacteria has arisen in several different lineages. We also understand that horizontal gene transfer is prevalent. This can create a mosaic genome of genes from other species mixed into one, and can also make phylogenetic placement very difficult. This phylogenetic tree represents our current understanding of the three domains of life: domain Eukarya, Archaea, and Bacteria. Note that all three domains share a universal ancestor, however, that universal ancestor is currently unknown. Within the domain Bacteria, we have gram-positive bacteria, as well as the gram-negative bacteria represented by Proteobacteria, Chlamydias, Spirochetes, and Cyanobacteria. The domain Bacteria is highly diverse and is estimated to contain over 700,000 to 1.4 million species. However, only about 16,000 of those species have currently been identified. There are many different nutritional modes, as well as many pathogenic and beneficial bacteria within this domain. It currently is represented by 5 major groupings: the gram-positive bacteria as well as the gram-negative bacteria, as stated previously. The gram-negative bacteria consists of Proteobacteria, Chlamydias, Cyanobacteria, and Spirochetes. Proteobacteria is a very diverse group that includes autotrophs, heterotrophs, pathogenic, as well as beneficial bacteria. Chlamydias are a parasitic group that primarily relies on an animal host cell for its resources like ATP and other nutrients. Chlamydias typically lack the peptidoglycan layer. Spirochetes are considered heterotrophs that possess a rotating flagellum-like filament for movement. Spirochetes can be free-living or pathogenic. And cyanobacteria are considered to be photoautotrophs that carry out photosynthesis. They can be solitary as well as filamentous and are common in phytoplankton. Cyanobacteria are an important group to consider, particularly when examining endosymbiosis and the development of eukaryotic cells. Gram-positive bacteria is considered to be a very diverse group that can include pathogens, beneficial species, decomposers, as well as other nutritional modes. These bacteria may be solitary or they may be found in colonies, and some species are considered to be important in the culturing of various types of antibiotics, such as streptomyces. There are many common pathogens that are considered to be gram-positive, and these include the familiar bacterial species such as Staphylococcus aureus, Bacillus anthracis, Clostridium botulinum, and mycoplasmas. We are now going to take a look at the domain Archaea. Many of these organisms have unique traits to the domain. However, some of these organisms will also share traits with either bacteria as well as eukaryotic cells. The domain Archaea includes a tax supergroup. This supergroup is important as they may shed light on the evolution of eukaryotic cells. The tax supergroup includes Thaumarchaeota, Algarchaeota, Crenarchaeota, as well as Coarchaeota. Organisms within the domain Archaea are known to inhabit both moderate as well as extreme environments. Some organisms are known to be anaerobic methanogens. This means that they can use carbon dioxide in order to oxidize hydrogen gas. They can use this to produce both energy and methane gas. This can include both extremophiles, like halophiles, that live in high salinity environments, as well as thermophiles that live in extreme heat. Pyrococcus furiosus, for instance, is one of these species that has specific adaptations that can stabilize proteins as well as DNA at very high temperatures. This has important implications, as this species can serve as a source for DNA polymerase in PCR reactions, because the DNA polymerase is already stabilized to withstand very high temperatures. Prokaryotes, such as those organisms seen in the domain Bacteria as well as Archaea, are known to serve very important roles in the environment. Many of them are considered to be integral to proper ecosystem functioning. This is because they are responsible for chemical recycling. Many species can serve as decomposers, returning important nutrients back into the system. This allows for these molecules to be uptaken by other organisms. This includes carbon and nitrogen fixers, oxygen producers, as well as some prokaryotic species that supply important nutrients such as potassium as well as phosphorus. Many species also have important ecological interactions, such as mutualism, commensalism, and parasitism. And finally, prokaryotes can have both beneficial and harmful effects to humans. In the instance of mutualistic bacteria, the human intestine can contain 500 to 1,000 bacterial species. This can help us to be able to produce as well as break down nutrients for absorption. However, there are also many pathogenic bacteria. Bacteria themselves can cause more than 50% of human diseases. This is mainly due to the production of both exo and endotoxins. They can be directly harmful, or they can also be carried by vectors to cause human infection.